All right, everyone. Thank you all for joining us today. My name is Stephen Prager. I'm the director of the Appleton-Whittell Research Ranch of the National Audubon Society. We are an 8,000-acre field research station and sanctuary in southeastern Arizona. And here we put science to work to answer the questions that are critical to the conservation of birds, wildlife, and the places that they live, with a particular focus on grassland ecosystems. We're really excited to be kicking off this webinar series today, to be giving y'all a behind the scenes look at some of the work that is underway on the research ranch this season. And I think most importantly, to elevate our really awesome batch of 2024 Appleton-Whittell Research Ranch Fellows. And before we dive in, I just want to give you a little bit of background about our fellowship program, what it is, what it provides. It was launched in 2010, and it supports up to 3 research fellows annually. Our fellows are young, between the ages of 18 and 30 scientists focus on conservation research that can be facilitated here on the Appleton-Whittell Research Ranch. Every fellow receives a $1,000 cash stipend to support their research needs. They can spend that on anything from field supplies to lab equipment to food to travel, whatever they need to make the work happen. And they also receive a $500 Appleton-Whittell Research Ranch housing voucher, which affords them up to about 2 weeks worth of overnight stays here on the research ranch. Also communication support from Audubon Southwest and our team here, evidenced by the webinar you guys are on today. If you're curious about applications for next year, the application window typically opens in early February, and applications are due towards the end of March. So now what you are really here for is to learn what our fellows have been up to this season. And for that, I'm going to pass it off to 2024 Appleton-Whittell Research Ranch Fellow Laura Nicholson. She is a PhD student in plant conservation, joining us from Northern Arizona University this year. So I will stop sharing my screen and I will pass it over to you, Laura.
Awesome. Thanks, Stephen. All right. We'll get this going here. See if I got this. Okay. The fun thing getting this up. Okay. All right, let me swap that over really quick. Let's try that again. Swap displays. Okay. I think you guys are all good. Hopefully seeing the right thing. Awesome. Great. Well, thank you so much, Stephen. And I am so honored to have been a fellow for this year. It has really been a wonderful experience, and I'm really excited to share a little bit about my research and the work that we have been learning so much about. So before I dive in, I just wanted to share a little bit about myself. I am originally from the town of White Bear, Minnesota, where my love for wildlife, trees, and the outdoors really began. I did my undergraduate degree in ecology and evolutionary biology at Rice University in Houston, Texas, where I started my senior thesis exploring the pollination of rare trees. Following this, I had the opportunity to work in a number of different positions, working with wildlife monitoring in Lassen Volcanic National Park in Northern California, studying pollinators in Ecuador and prairies in Western Minnesota before I had the opportunity to do my master's in wildlife ecology and conservation at the University of Florida, where I was exploring the effects of hydrologic restoration on an endangered bat species. We did actually just get an article published on this work if you're interested. Big picture, we did find that hydrologic restoration is good for bats, so that was good news. But following this, I worked as a data analyst for Fish and Wildlife for a year before starting my PhD here at Northern Arizona University, where I am now exploring questions related to the famous Emory Oak. Broadly, I'm really interested in conservation ecology, landscape ecology, plant-animal interactions, and restoration ecology. So here is a quick outline of what I'm planning to talk about today with you all. Firstly, we'll talk a little bit about the history and ecology of the Emory Oak, followed by some threats that are currently facing this important species, and then we'll talk about some of the research projects we're currently working on. Then we'll finish up with some future directions and conservation implications. So the Emory Oak, or Quercus emorii, is a keystone species here in the Southwest, and it is really important for both ecological and cultural reasons. This tree was originally named for William Helmsley Emory, who was a Civil War era surveyor and cartographer and civil engineer who played a significant role in mapping and protecting settlements out west, which undoubtedly came with a conflicted history. The tree was named for him to recognize his contributions to the Western understanding of the biology of the area and for being the first Westerner to allegedly discover the species. However, of course, this tree was already deeply known and cherished by the Western Apache people for millennia and is known as Chichil in the Western Apache language. It is actually really amazing to see how the cultural connection plays out between this species and the Apache people and how this had deep ecological effects. If you look at this map here, you can see how the range of the Emory Oak is tightly connected to the range of the Western Apache tribes, and there's a lot of evidence that there was likely human-mediated dispersal of this species when tribes were established in the area, in addition to communities thriving where Emory Oaks were living. And this relationship was really based on a practice of acorn collection. But this connection between the land and the people was really disrupted during Western expansion with the removal of Native peoples from their traditional homelands onto the reservation system. And land ownership and regulations have been a significant barrier to the traditional practice of acorn collection ever since. A little bit more about why Emory Oaks are so important to the Apache people. This practice of acorn harvesting has been a longstanding practice and source of sustenance for people. And as noted by Vincent Randall, who is the late cultural director and tribal elder of the Yavapai Apache Nation, he noted that Emory Oak are vital to almost every Apache social and ceremonial function, as well as being important indicators of environmental health for these people. The acorns of the Emory Oak are really cool because they are so low in tannin content, which means that you can eat them directly from the tree without having to process them. They're really low in that bitterness. And so there's a strong tradition of consuming these acorns across the range in Arizona, New Mexico, and across Mexico. In Mexico, they are known as bellotas. And so traditionally, the acorns are either eaten raw or ground into an acorn meal and cooked into a stew, oftentimes with meat and corn. And there are a lot of recipes online that you can look up and read about to use these acorns, which is really pretty cool. So beyond the general interest in this tree due to its cultural importance, there's actually particular urgency to study the species due to recent concerns about its long-term viability. Recently, tribal elders have noticed consistent decreases in acorn harvests and reached out to researchers at NAU to investigate the health and longevity of this species population. This has resulted in several large-scale projects, including the Oak Tree Project, which is the Emory Oak Collaborative Tribal Restoration Initiative, which is headed up by my advisor, Dr. Sarah Souther, and is looking to explore the effectiveness of different management interventions to restore Emory Oak habitat, as well as the Dices Project, which is a huge collaborative project involving over 12 different researchers investigating different aspects of the species, from demography and ecology to dendrochronology and genetics to understanding social barriers to acorn collection. So I feel very honored to be working on this project. So now that you have a little bit of background about the history of this species and why it is so important from a cultural perspective, let's discuss a little bit about the important role it plays in the ecosystem. It is one of the dominant oak species in the Madrean Sky Islands and oak woodlands and savannas across Arizona and New Mexico. And the species plays a key role as a soil stabilizer as well as a refuge for wildlife. It not only provides sustenance through nutrient-dense acorns, but provides habitat and nesting locations for a wide variety of birds and iconic Arizona fauna such as the coati, the mountain lion, and javelina. The Mexican jay is a particularly deep connection with the Emory oak as it is a primary disperser of its acorns. So as you can see from this map, the range of the Emory oak extends from the Sedona area down into Mexico, and from over in the Prescott area into New Mexico, and even ranging on the Texas border. It's mostly in North American Madrean oak woodlands and savannas, and ranges from about 4,000 feet to up to 7,000 feet in elevation. It's often found associated with alligator juniper, pinyon pine, and ponderosa pine in forests. So we do see some interesting regional differences in stand characteristics for the species that I want to point out, which is something that we wonder if it's playing a role in terms of differences in acorn success, as well as recruitment across the range. So in the northern region, in the Payson and Prescott area, generally the trees are a little smaller, more shrubby, and closer together within sites. And we seem to be seeing anecdotally that there's more acorns and higher recruitment as compared to the southern region, such as on Appleton. We are seeing these larger, older trees that are in kind of more the savanna ecosystem type with a lot fewer acorns and lower recruitment. So that's something we're interested in exploring further to see if there are these regional differences in success. From an individual tree standpoint, we see trees ranging from 30 to 60 feet tall in height when mature. The leaves are dark green, waxy, and holly-like in shape, as you can see from the top right corner picture. The trees are actually drought deciduous and mostly evergreen, which means that most of the year they keep their leaves, but during periods of drought or during the dry season, they'll drop those leaves in order to conserve water, and is a drought related adaptation. The acorns themselves are green when immature and then become reddish brown when mature, and they have this kind of oblong football shape and are relatively small. As you can see on that photo, comparing across different acorn types, they're in the top right corner and are really pretty small relative to other acorn sizes. In terms of timing, they mature in late July and August following the start of the monsoons. One note in terms of identification, the Emory oak is really pretty distinct from a lot of other oak species here in the Southwest. It most closely resembles the Arizona white oak. And so we have some tips and tricks to identify between, or just differentiate between these two species. So from a bark standpoint, you can see that the Emory oak has this darker, chunkier bark as compared to the white oak. And from the leaves is really kind of the best way that we can differentiate. The Emory oak has this shiny, waxy underside, and whereas the white oak, if you flip it over, it has this fuzzy matte finish. And we do something called the dust test, where if you rub your finger along the underside of the Emory oak, there's a little bit of dust that will come off, and it has that shiny surface. Whereas the white oak, if you run your finger along it, the dust does not come off, and it maintains that fuzzy finish. And so you know that is going to be a white oak. And that holds from little tiny seedlings all the way up to these large adults. So now that you know a little bit more about the biology of this species, I want to touch on some of the factors threatening its survival. This kind of breaks down into some main groupings with abiotic factors, non-living, such as fire, elevation, climate change, and drought, and then the biotic factors such as disease, wildlife, weevils, livestock, and humans. As you can see, many of these factors also influence each other in addition to the Emory oak, which can lead to a very complex web of interactions. And this diagram here, you can see the positive is what we understand to be a positive influence on that other factor. The yellow is kind of a mixed influence, and the red is a negative impact. So as you can see, it's a very complex system that we are looking at. So now on to our research projects. So firstly, we'll be looking at the population dynamics. Each of these are a factor that is playing a role in my PhD. And so the first one we're going to talk about is the population dynamics and drivers of germination. So for this project, I had two main questions. I firstly wanted to understand what factors drive Emory oak success across the landscape and whether drought plays a role. And secondly, to understand what site factors predict survival of the germinants. To do this, we have set up 100 Emory Oak plots across the range in Arizona and New Mexico. On the map to the right, you can see the orange dots indicate the locations of our plots. And for each of these plots, we assessed the general plant community as well as taking tree-specific measurements and assessments of the health of the trees. This plot here, there's a diagram there that describes the data that we would take at each plot, including the community metrics, including line point intercept, to understand the understory plant functional groups that characterize a plot, whether it's, grasses or shrubs or other forbs, as this can oftentimes influence whether a germinant of an Emory oak is able to be successful or not. As well as belt transects to understand what kinds of other tree species are successful there and are associated with Emory oak success. And then on the left, you can see what one of our plots looks like. For each plot, we tag the location of every adult and juvenile Emory oak, which, as you can see by the picture of the example tagged photo down there with the little coin, we would call them little necklaces for the trees. It was sometimes a lot of work to find every single one of these germinants within all the grasses and shrubs of the plots. Once we flagged all of these plants, we would then use a very precise GPS to record the locations of every single tree and seedling, and then input that into an online GPS format. So you can see what that looks like from a map standpoint. This was a really powerful way of collecting data because it would allow us to connect the locations of the data and the proximity of other trees to all of that size characteristic data that we took as well. So for every Emory oak, we would take the size of the width and height, as well as the diameter of the stem, as well as looking at different health metrics. So that all in all, it was a lot of work. And then in addition to that, we'd also look for evidence of livestock, look at the soil conditions, and then counting the number of acorns on the mature trees. All this data allowed us to start teasing apart what might be driving germination success. To do this, we basically took all the data, put it into spreadsheets, and then did a bunch of math and statistics in order to understand the patterns that we were seeing in the field. We first started with data exploration. This involved looking at different visualizations of the data in different ways to understand what might be driving the success of the species. This data is very preliminary because we just finished collecting our second year of data just a few weeks ago. So these data are looking at just our first year of data back from 2023. So the plot that you're looking at here, that's a bunch of dots and lines, is a visualization of a principal component analysis, or PCA, which basically takes all the data we've inputted and put it into multi-dimensional space. And then it draws connections between the factors to help us understand what might be driving our result factor. So what might be driving the success of the germinants, or number of germinants that are successful. So from this visualization, we can see from the length and direction of the red lines that geographic location, so latitude and longitude, human populations, altitude, and precipitation may be some of the most important influences on germination rates. So with that in mind, we then conducted generalized linear mix models, which allows us to predict what factors may have important relationships with number of germinants at a site. And so from these analyses, we found that elevation, as well as factors related to wildfire, seem to have an important impact on how many germinants one site may have. So from elevation, we saw that there's a positive relationship. So as we got higher in elevation, we saw greater numbers of germination, so higher rates of success in reproduction for the Emory oak. And for fire as well, we also saw that there's kind of a complex relationship with fire. It seemed that there was differing fire sizes, like a larger fire size seemed to have a positive relationship with number of germinants. And so that is basically indicating to us that fire is an important factor that we need to explore further. The other factor was a wildfire risk score, So that was a little bit more complex, but basically just indicates that fire is important. And seems to have a positive impact on Emory Oaks, but can also be negative depending on the situation and severity of the fire. It was interesting to see that drought did not come out as an important factor as a driving force, but that's something we'll discuss further. From looking at those results, we have some limitations to them. As I mentioned, it was based only on year 1 data, and so we still need to incorporate the next year of data that we have collected. The drought factor, the drought designations for our sites were based on hand-drawn maps from a plane, so we wonder if incorporating other sources of data that might be better describe drought might better inform that relationship. We are also going to continue to monitor growth and kind of see how that influences the survival of those germinants in addition to just their presence. And then something we're looking forward to looking into further is looking at germinant survival based on drip line theory, which is basically the idea of where a germinant is able to grow, whether that's going to allow it to be successful. This conceptual diagram here kind of indicates where an Emory Oak germinant might be happiest. So it's, does okay with a lot of cover, it very close to the tree, and we think it might do best underneath the outer drip line of a canopy because the water falls from the sky onto the trees and goes to the kind of the outside based on where the leaves are. It falls in an outside ring so the germinants on that outside ring might be the most successful, whereas the trees, the Emory Oak seedlings that are just out in the open might be too desiccated by the sun. So that's something we're excited to look into further. So now moving on to looking at disease ecology, which is our second factor that I'm really interested in exploring. This is really based on the disease triangle that I'm sure is something you guys are all familiar with based on our experience with the COVID pandemic. But this is something that plays a role also in tree disease dynamics, where you're looking at the host, the pathogen, and the conducive environment. And you need all three to be favorable for a disease to take hold. So in the case of the Emory Oak, we have the susceptible host, which is the Emory Oak. We have a conducive environment, which is exacerbated stress for the Emory Oak through drought and elevated temperatures. And then we have our main disease of interest, which is Biscogniaxia, which is a pathogenic fungi that has been recently understood to be of particular concern for the Emory Oak. And this is something we're going to talk about further. So Biscogniaxia is this fungal pathogen that has these fruiting bodies that push out the bark on an Emory Oak. Here's an example of what this kind of looks like. It has this pimple-like parathethia, which is basically what is releasing these spores that infect the Emory Oak. There's kind of some interesting ways we are oftentimes out in the field looking to assess the health of Emory Oaks. So we oftentimes are trying to understand where there's, where is there Biscogniaxia, where are there injuries, or is the tree totally fine? And it's something that I've learned takes a lot of experience and practice. So as you can see here, you can kind of try on your own. These are pictures in the field of when we get out there and we say, okay, is this a disease or is this just an injury or is this just, the tree with a little bit of sap? And they can oftentimes look pretty similar, and it takes oftentimes a trained eye to really assess how the tree is doing and what is Biscogniaxia. So you can take a look here, look back here at these photos, kind of look for that parathethia. You can try your hand at identifying, whether 1, 2, 3, 4, 5, or 6 might be Biscogniaxia. People are welcome to put into the chat which one they think might be Biscogniaxia, or you can keep in your mind if you're not sure. We'll give people a few seconds here to think about it. Chat's disabled. So here we can see if you were correct. The first one was indeed Biscogniaxia. The second two were not. Number 4 and number 6 also were. And the other ones, these are just stains, and this one appears to be kind of a sap that is coming from the tree. So as you can see, it can be very challenging to try to identify those. So a little bit about the biology of Biscogniaxia. This pathogen is opportunistic. So it basically has two types of life stages, where firstly it's an endophyte or endophytic, meaning that it just lives on the tree. It isn't harming the tree, but it's also not benefiting the tree. And this is kind of its long-term status. It's not totally clear how long the Biscogniaxia can live in the tree in this stage, but it appears to be triggered to transform into a pathogenic aggressive fungi through tree stress, as you can see by this little diagram here. It's kind of like when children or college students or just anybody, when you are stressed and lacking in sleep, you can sometimes turn into a little bit of a monster, and that is basically what happens to this fungus when the tree is under either heat stress or water stress. It triggers this transformation, where the fungus will invade the sapwood and cause a lot of damage for the tree. And it's a little unclear in terms of timing, how long it takes for this that to kill the tree, whether it's a few years, many years, and that can also depend on what kind of defenses the tree has and how stressed that tree is. It appears that this fungus, we don't see vertical transmission, which basically means that we aren't seeing if an acorn has this endophyte, it doesn't seem to transfer to when it's an adult. So that is good news. It seems to be overall an adult disease, as we have found from our preliminary data. It really doesn't seem to infect the juveniles and seedlings, but mostly older trees and dying trees. But the thing that is of concern is that we are starting to see it in healthy trees, which is maybe an indication that a lot of these trees are under a lot of stress. So some signs and symptoms of Biscogniaxia in Emory oaks. And one quick note is the difference between a sign and a symptom. This is something I didn't really know about before I got into this, but it's an important distinction within tree pathology. A sign is basically a direct evidence of the disease itself, whereas a symptom is something the tree is doing in response to the disease. So some signs or some symptoms of it is on the leaves is a discoloration of the leaves. You'll see the tops of branches starting to die. You can see a picture of the discoloration of leaves here. It's a stem and bark disease primarily, so you'll see or the inner bark and xylem will have some necrosis or death, and then you'll see the sign of the disease, which is the charcoal canker itself, which is basically that lesion pushing out the bark of the tree. And that's where the spores are released. Systemically, we see invasion of the sapwood, which cuts water supply to the canopy, and just overall tree decline, which is the biggest symptom of this disease. It also seems to increase susceptibility to attacks by other pathogens as well. So there's a couple different ways to try to sample for these fungal pathogens. The three main ways, firstly, is to manually go and sample those trees, go out to the field and look at them and look for those signs and symptoms of disease. This can be then taking biotic samples of the lesions themselves. You can also take twig samples and core samples of the trees to then plate out those endophytes to understand if the Biscogniaxia is there. You can also mark the samples on the trees, those lesions, and track that over time to see how the lesions are changing and what other signs and symptoms change over time. Secondly, you can look at genetic analysis, again, taking those twigs and leaves. And then there's also air spore sampling. Here you can see a diagram of those air spore sampling setups and what that kind of looks like. So there are a lot of pros and cons to these different sampling methods. The manual review and sampling of trees is what we have been doing. It's very time consuming. It just takes a lot of time getting to sites. As you can see from our map here, we have sites ranging all around the state. And this is not a scientific diagram here, but we basically see, oh, sorry, have these been up here this whole time? My apologies. Let me move that out the way. Hopefully that's not in the way for people. there is a lot more driving that goes along as compared to sampling when it comes to trying to sample for these sites. And it can be really difficult to measure and assess the canopy of these species because these trees can be so tall. Again, they can grow up to 60 or 70 feet. So if there is a Biscog sample way up in the canopy, it can be challenging to see that in the first place. And then a pro of the sampling method is that you can get really good quantitative data on growth and the impacts over time for individual species. So it's kind of this really granular data that you can get a lot of information from. Some pros and cons from genetic analysis. It can be really a fast way to identify species and can be much more accurate than trying to do it morphologically. However, it can be very expensive and there can be difficulty with developing primers to process that data. As for air spore sampling, this is something that's also very time consuming from a field and lab work standpoint, but it can allow you to have real-time monitoring of what is basically in the area, not only on those trees, but in the air, and can be pretty cost effective when it comes to trees. Yeah, this is just a photo of some of the challenges that come along with field work. Car troubles is a very common predicament. Even though you think you're being out in nature, a lot of times you're just driving. So what is happening with Biscogniaxia and the Emory Oaks here in Arizona? These are some of the questions we wanted to understand. Was basically, where is it? What kinds of trees is it infecting? Is it dead trees, alive trees? What are the ages of these trees? And can we predict where we are going to see infection? And finally, is it getting worse over time? So this plot here shows all of our plots and where we see Biscogniaxia. It's interesting to see that it is kind of all over throughout the range. We predicted that we were going to see it really down in the southern area because those are areas that are more affected by drought. And we had noticed anecdotally that there was some cases of disease there. But once we started really looking for it, we found it throughout the range, which was really interesting and a little concerning to see because it was only about 5-10 years ago that this disease was first described here in Arizona as affecting Emory Oaks. So we are learning that it is much more common on adults and dead trees, but it is also infecting both adults and juveniles, live and dead. And that is across the range. That is really pretty concerning. We saw infection at 16 of our 100 sites, which doesn't seem like a lot, but considering the rates of infection is pretty concerning. Some other follow-up questions we had is which species it actually is. We are assuming it is Biscogniaxia mediterranea, which is the same species that affects oak trees over in Europe, and it's a really big problem for the cork industry, especially in Portugal. But we are interested in investigating if it is an entirely new species, whether it's a native species or if it's invasive or exotic, and just diving into those questions further. This diagram here, this map, indicates the infection of trees, both live and dead. So you can see that it is the dark ones that are tan, those are infecting the dead trees. But it's very concerning to see that the yellow here is where it is infecting live trees in addition, because it is usually understood to be something that affects the dead trees and is kind of a sign that the tree is dead or dying. But the fact that we're seeing on live trees is also very concerning. There's also something interesting going on here where we were wondering if there are two different species entirely, because we are kind of seeing this infection in these clusters of a southern and northern population. So that's something we're interested in exploring as well. It's overall, we performed similar analyses to the study we did previously with the number of germinants. This was using generalized linear mix models. And so we used our data to try to understand what might be allowing us to predict infection. And the main thing that came out as important in predicting this was crown dieback, which makes a lot of sense because this is known as a sign of tree stress. And so that fits with the current literature that, you know, when you see a lot of crown dieback or the tree is dead, there is a high likelihood that that tree is going to be infected by Biscogniaxia. So we have so many more questions than answers. We also are wondering, you know, how it is transmitted. Is it mainly through the air, or how the other trees are maybe playing a role in transmitting between species, whether insects are playing an important role in transmission. So there is so much more to learn on that. And then there's a whole matter of treatment and what we can do about Biscogniaxia. So there's a lot of different factors, and it really depends on the scale on which you are looking at this feature or at this problem, really. So at a tree level, there are antifungal injections for certain funguses, but there's not currently one for Biscogniaxia, but it's something that it could be worth exploring. It's possible that trimming dead branches could help with controlling this disease. At a stand level, it appears from the literature that prescribed fire can have a really positive effect in controlling this disease, as well as thinning and controlling that scale of density within a plot. And then at the landscape level, combating climate change, which, it's not something we're going to solve next week, but it can play a large role in affecting the amount of stress that these trees are under. So as I mentioned, climate change is a global problem. It's very complicated, but supporting decarbonizing policy decisions and environmental water rights can go a long way in helping this tree species long term to combat this disease. When it comes to fire, using fire can be a really effective way to treat this, but there are smoke and land use considerations, and there's considerations when it comes to managing wildfire versus prescribed burns, and kind of having that wildfire human interface. And then thinning can also reduce density and reduce competition and thinning and transmission of the disease, which can be helpful, but does take a lot of time and money, and stumps may be a source of secondary infection if that dead tree is removed, but it has that disease in the stump. That could be a source of transmission, so something to consider as well. You can see here from this photo what it looks like for the injections to go into the tree, but that again is something that's very expensive, has not been developed yet for this disease, and really is kind of something you do on a tree by tree basis. So yeah, again, the injections, it can be practical for individual trees of cultural importance, but there's a lot of ethical considerations with this true, especially when it comes to working on tribal lands, making sure that tribal opinion is taken into consideration and how this might impact the acorns of the trees to have that fungicide. And again, there's nothing currently available for this species. But there are some preventative actions, it seems like, that could have a positive impact on protecting this species, such as pruning younger trees and removing dead branches properly. And if you're planting Emory oaks or other tree species, having a lot of forethought about which species you're planting and what density you are planting them. This figure here just describes a little bit how branch removal can be done correctly, because if it's incorrectly done, it can expose a tree to even more infection, but if it's correctly done, that can reduce infection as well. So a little bit more about those tree level fungal treatments. So as I mentioned, there's not yet a fungicide available to combat Emory oak or this disease for Emory oaks, but this is something we are actually looking into from the lab standpoint. We are trying to explore any potential antagonistic relationships between Biscogniaxia and other endophytes. And so to do this, we have taken twig and core samples from sites infected with Biscog, and we're basically trying to identify different endophyte species and Biscogniaxia. From this photo is a picture of some of our plates where we have grown out these different fungus and bacteria from the tree species. And you can see there's a high diversity of species across that are just living within these Emory oaks, which is not too surprising when you think of the keystone species that they are. But we basically grow out these endophytes, and then we are going to do genetic analyses to understand what species they are, and then we would put them on plates next to each other and see which ones are preventing the growth of Biscogniaxia. We are then going to use that information and apply it in a greenhouse setting to understand how the microbial community might be playing a role in disease resistance for protecting the Emory oaks. So to do this, we are going to be using soil that has positive has a microbiome intact, and then soil that we have sterilized, and then basically putting Biscogniaxia, inoculating seedlings of Emory oaks that we have grown with Biscogniaxia and then other ones without, to try to understand how the presence of this inoculated soil may help defend against severity of disease by Biscogniaxia. So it's a little bit complex, but this basically shows the design of this experiment that we are going to be conducting to understand how this might be affecting disease resistance. So this is a whole process. Just give you a peek into the scientist's day in the life for doing this project. We basically collected acorns from the field, sterilized the soil, did some floating and spoon testing, which I'll talk a little bit more about. Then we'd sterilize the acorns, add soil, and plant them. So currently we have over 1,500 Emory Oak seedlings growing for this project, which is very exciting. And we're learning a lot just about Emory Oak germination in the first place. For example, the classic float test, which is basically where you put the acorns in a bucket with water to determine whether they are viable or not. We found this is actually inaccurate for Emory Oaks because almost all 99% of our acorns did float, which would have normally indicated that they were not viable. But because they all were floating, we decided to plant them anyways, just in case. And we actually found over 99% of them grew, even though they had failed the float test. So that was really pretty informative and interesting to learn that. It is something that we will be writing up. But then something that did fit with conventional wisdom was we did see that the larger the acorn, the more likely it was to germinate. So these really small acorns were usually ended up being either infested with weevils or were just not viable acorns. And that was something we were able to test through what we called the spoon test, where we would basically take a spoon and apply pressure to the acorn. And if it immediately crumbled, we knew that it was not going to be viable. But if it was a strong, healthy acorn, then we were able to plant it. So from all this, I know this is a whole lot of information here, but we are basically learning that the Emory Oak is experiencing these novel environments with climate change, which is driving these new novel interactions with the disease. There are so many different ways to monitor and treat disease, but there's a lot of ecological and cultural considerations. And hopefully you can see that, research can be really applied and we can learn a lot to inform management through that. And so a last little bit here, I just want to touch briefly on this final project that's at the beginning stages with wildlife interactions. So as I mentioned earlier, the Mexican jay is a key seed disperser for the Emory Oak. So they are basically taking, these acorns from the trees, and a lot of times it's really pretty interesting. They're corvids, they're so smart. They are able to determine which acorns are the best by weighing them. And there's a lot of really cool work that I have resources about if you want to read about that. They are weighing them, choosing the best ones, and then they are stashing them in favorable locations, which they obviously don't remember where all of the acorns they place are, and the ones that they don't go back to retrieve later then are effectively dispersed by their actions, and oftentimes into novel environments that are usually suitable, so they can be really powerful reforesters of this species, which is really pretty cool. So for this project, we have game cameras that we have set up, and we'll be setting up additional ones. I have two primary questions that I want to be asking, which is basically understanding how the presence of predators in Emory Oak stands might be reducing consumption, but maybe also dispersal of acorns, and whether there are differences in the response of acorn consumers and dispersers to different types of predators. So those are my two main questions, and this is a particular interest as we see Mexican wolves, reintroduced into this area and what role they might be playing in these ecosystems. So far, we have 18 cameras placed in the Sedona area, and we have additional 18 that we are going to be putting out this year. And this is contributing also to the Snapshot USA program, and we'll be using AI to analyze these photos for to understand these wildlife communities. But these are still really in the beginning stages. So I'm really interested to know kind of what you as a birding and wildlife and conservation community want to know in terms of wildlife interactions within the Emory Oak community. And so please feel free to reach out. My contact information should be available, and I will be able to send that out. So via email, I would love to hear your thoughts and what you kind of want to learn about within the realm of this field. So on some final notes, when thinking about conserving the Emory Oak, what role can the individual person play in this field and in this problem? There's kind of three main ways that you can play a role, and that's through awareness, advocacy, and support. So in terms of awareness, just to keep learning about these conservation issues is really a really powerful way to better inform yourself and inform others on kind of what the current status is and what can be done to help. Respecting Native people practicing traditional acorn collection is really great if you live in the Arizona area. Oftentimes people will try to help by bagging acorns, but that actually can be bad as the acorns go bad very quickly. And so it's much better to just leave the acorns out and, just let people know that people are welcome to come on your land if you're okay with that, to come collect acorns. And just learning more about that traditional practice, and just inform others about this important practice and about respecting Native rights to collect those acorns. Then advocacy. Supporting bills that protect public lands, wildlife, watersheds, and Native rights can be a super important way to make a difference in our state. Writing to your congressmen and congresswomen and your representatives to advocate for causes that you care about, such as this. And then finally, supporting organizations like the Audubon with your time and resources to protect these ecosystems and support research that can contribute to conservation. Also volunteering for research projects. Scientists oftentimes, almost all the time, need a lot of help and are really willing and open to have help out in the field or in the lab or in the greenhouse, trying to get these projects done, because they are oftentimes very large scale. And so we love working with members of the public and people just interested in learning more about research. And you can also be a citizen scientist. If you haven't heard about iNaturalist, really encourage everyone to explore that, and the app called Seek, which can help you identify different species that you see out in the field. And then by posting it to iNaturalist, that can help a lot of scientists who are trying to understand the distribution and health of species across their ranges. So that is a really powerful way to contribute as well. So here are just a few resources that we can distribute as well, just to learn more about the Emory Oak and its cultural history, seed dispersal by these Mexican jays, and the current projects that we are working on. And so with that, I'm going to open it up for any questions you guys may have. I've really enjoyed being able to talk to you guys a little bit about our research and the history of the Emory Oak and why we should, be protecting this really important species. So thank you all so much for listening and for your time being here today.
Thank you so much, Laura. That was fantastic. The Southern Lab has always been such a pleasure to work with, and you are no exception. And I don't think I could have designed a research project that better ties together all of Audubon's priorities in this region in such a compelling package. So thank you so much. Everyone on the line, if you want to pop into the Q&A and drop questions in there, I'll read those out loud for Laura to respond to. You can also raise your hand and I can open up your microphone if you would prefer to ask a question that way. So I'll just start from the top here. The first one, Laura, Suzanne, she is curious if you measured actual precipitation on your plots or if drought was a derived variable from other factors.
Yeah, the drought is a super tough one. That's something that was derived. It was based on maps that were hand drawn from a plane, which is wild to me that is the way that we are doing this, but it's hand drawn from a plane looking out the window at, greater areas of where they're seeing, death due to drought. And that's something that's conducted by usually the Forest Service, but is also conducted by other research groups. There's also lots of other ways to look at drought. So like, as you mentioned, precipitation, that's not something that we have been collecting like at our plots, specifically because they're really spread out and hard to revisit. So that's something that can be very costly over time, but there's a lot of different maps and that are available through NOAA and through other online sources that kind of are tracking drought over time and have these different metrics that are looking at the meteorology of these different areas and can get really fine scale, which is really pretty powerful. So we're looking at kind of incorporating a lot of those. One of them is called BioClim that incorporates like 18 different metrics, looking at the atmospheric changes and over time and those characteristics of the site. So we're interested in looking at that more and kind of using different sources to describe drought and lack of precipitation in areas.
Let's see what else we got. Is there anywhere, this is from Nicole Gonzalez, is there anywhere that folks can report suspected Biscog sightings for review to help with mapping efforts?
Yeah, that's awesome. And I wish there was like more of like a better like app or anything like that, but I think you can post like sightings on iNaturalist through using, you take a picture and you can note that you've seen it there and so post it on there, as well as you can also report it to the Forest Service. There's a whole disease pathology unit that is really that we are collaborating really closely with, and we are really interested in any sightings that are seen. And so you can report that to the Forest Service and they'll pass that information along to us. So yeah, and also just reach out to me. Let me know if you see any. We're really interested in finding more areas and kind of tracking that over time.
Cool. Kind of a related question. Have you found Biscognoxia on the Appleton-Whittell Research Ranch?
Yeah, so we've been looking around for, and there we have seen evidence of it, which is definitely, it's in the area. It's definitely of concern. And so that's something we are going to be tracking over time and kind of seeing how that changes. And if we're, seeing greater rates of infection, we have several plots on the ranch and ones that are nearby there. So we can kind of continue to monitor that over time and share that data with you guys.
Cool. I'm going to kind of bounce around here, but there's kind of a related question, and I think I'll take this one. Has the research ranch changed any land management practices based on Laura's research? And the short answer is not yet. But in the next couple of months, we're actually going to be meeting with a lot of the members of the wildland firefighting and science communities to start kicking around the idea of prescribed burns here on the research ranch. And that conversation is definitely one that we're going to loop back to Laura and the Southern Lab with.
Yeah, we're still in the preliminary stages. So we're researching this a while.
This one's from Tucker. Excellent work. Have y'all considered any sort of like compost or compost tea kind of a thing to improve local soil conditions around individual trees as a way to push back against this fungus?
That's a really interesting idea. That's, yeah, not something I've thought about, but maybe that's something we could explore further. We've kind of been looking into the possibility of kind of a treatment by using other endophytes. But in terms of, it's one of those issues of scale. So, yeah, especially vulnerable sites might be worth exploring further in terms of either injections, but maybe some kind of, compost tea could be effective. That's kind of what we're trying to explore with this greenhouse study to understand if the microbes in the soil will transfer over to defenses in the tree, or if the microbes have to be already in the tree themselves, and how that microbe transfer is happening, if it's something in the air, if it's something mediated by insects, or if it's something absorbed from the soil. So I think that is something we'll learn more about as this greenhouse study progresses. And that is something that, might be really interesting in terms of applying that with a compost tea. So excited to explore that further.
I hope there's a really fun answer for this one. Do you need to climb the trees to get evidence of fungus in the tree canopy?
Oh my gosh, that is a very real thing. And sometimes there is really some issues with that. Thankfully, we've gotten around that because we are oftentimes hiking out to those sites. So tree climbing on top of that could be a lot, but we have these pole pruners. So basically there's like really long like painter's poles that have little snippers at the top. So thankfully when we take these core samples and twig samples, we're able to take those from the canopy without having to go up there ourselves. But oftentimes we do have our binoculars and are looking up at the lesions and we're like, we'd love to get, a lesion sample, but usually we just stick to the ones that are on the main bowls or reachable. But sometimes we do get up to some wild antics trying to get our samples, which is a little bit crazy. But no tree climbing for me so far.
Ah, cool. For this next one, I think I wish we had our last fellow from your lab on the line. But can you say any more about the likely relationship between predators and the success of the Emory Oak?
Yeah, so I know, yeah, Adriana, her work was super applied with this and it was really, really interesting and kind of sparked us wanting to continue her work and further expand it. But we really anticipate that predators have kind of a mixed relationship because they're likely reducing consumption of the acorns because the primary consumers of acorns like javelinas and doves are likely going to be kind of within this landscape of fear. Basically they're aware of the predators, are going to be consuming less and spending less time at the Emory Oaks because they are going to, need to run away from the predators. So we think there will be less consumption of the acorns, but we might also be seeing reduced dispersal if the jays are also, aware and spending less time by the Emory Oaks. So that's something we'll learn more about as we go along.
Cool. How long does an oak tree live once the disease is noted?
So that is a really good question, and one that we don't have a very good answer to. It's one that because this disease is really pretty novel and is not one we have seen and documented for very long, especially for the Emory Oak here in Arizona. We don't really know. We think it might be a few years, but it really depends on the severity of the disease, how stressed the tree is, and what other factors might be playing a role, such as heat stress and water stress, and kind of, whether it's the biscog that's, directly killing it, or if it's just really stressed and that's a sign of the stress. So it's a really complex relationship that's can be really hard to understand and unravel. And then there's the whole question of like, when is a tree dead? That is a whole other conundrum that is a surprisingly hard question to answer.
Fair enough. There's two more here. Good ones. Can collected acorns distribute pathogens if they're not sterilized before planting?
Yeah, yes and no. That one is um that one's complex. We do sterilize before planting just to reduce the risk. I think that's not something I like have a ton of experience with, or not something we've looked at too deeply, but there is the possibility of transferring those pathogens, and it is a definite risk. And so that's why we kind of take that precaution of sterilizing the surface of the acorn before planting. So that is a consideration, but is not something I know a ton about.
Get to play it safe, I suppose.
Yeah.
And this is the last one, and it's a fun one. What is your favorite acorn recipe?
Ooh, man, the issue is that all the acorns we've been collecting, we've been needing to use to plant. So I unfortunately have not gotten to collect enough acorns to be doing all the recipes that I want to make, but I have a list of all the recipes I want to try. So mainly I've just been stealing a few to just eat in the field when we're out there. But I have dreams. I have gotten to try the traditional Apache stew recipe that it's really very good. And so that's based on the acorn meal, and then it has, meat and vegetables. And so that is really delectable. I love baking personally, so I'm really excited to incorporate acorns into some of my baking recipes. I've seen some really cool recipes for acorn bars, which I think could be really kind of fun and kind of replacing peanuts in that role. So I'm excited to explore them further once we don't need to be using them for the greenhouse project so much. So I'll have to get
Very
back to
cool.
that.
Right on. Well, thank you again so much. And thank you everyone else for being here. Before we close out for the day, I just want to remind you that we will have 2 more of these webinars coming up. In October, we'll be hearing from Emma Sudbeck of the University of Arizona and her work to help us better understand one of Arizona's most problematic invasive species, the American bullfrog. And then on November 6th, we will have Marcus Williams, all the way from Virginia's Christopher Newport University, here to talk about how, the hard one to summarize in one sentence, but how climate and changing temperatures and animal physiology and behavior all come together in one of the ranch's most common reptiles, the ornate tree lizard. So I hope to see you all there. Thank you all again for joining us today, and have a great rest of your afternoon.
Wonderful. Thank you all so much.